Hello, welcome to the another new topic regarding bioinformatics. And the today topic is GENBANK. So in the last lectures, I just told you about the what is the bioinformatics and its applications, and what are the different types of the biological database. Biological database that deals with the protein information and the acid informations in the form of sequences and the structures which are obtained by different scientists all around the world. GenBank is a nucleotide sequence database and it is a primary database. So, in order to have the deep insight on this very topic, let us share one PowerPoint for which you can understand the topic very easily. In normal way, the bank that deals with the certain stock center and the gene that refers to the gene, and you know that the genes are made up of the nucleotide sequences. So these nucleotide sequences are nothing but it is the alphabets that are used to designate any sequences, and these sequences are written in the form of the A, G, T depending upon the what is the nature of the genes. So it was the 1965 when the first major bioinformatics project came on, and it was undertook by Margaret Dayhoff in 1965, and she was declared as the first lady who just developed the protein sequences database, and that was called as Atlas of Protein Sequence and Structures. Then after in 1970s, in early 1970s, you can say, in the Brookhaven National Laboratory, it established the protein data bank, and in this data bank, all the three-dimensional protein structure was submitted. Later on, in the next half of the 1970, it was the Needleman and Wunsch in 1970 who just paved the way for the routine sequence comparisons and database searching practiced by modern biologists. And in 1980s, the gene bank was established by the William Pearsons. And in 1999, all the universities they were just connected to the World Wide Web, via which now they are searching all the sequences that were submitted in the gene bank. What is gene bank? It is nothing but it is a complete collection of the annotated nucleic acid sequence data for almost every organism, sir. And the content of this gene bank that include the genomic DNA, messenger RNA, cDNA, expression tag sequences, then how through high throughput raw sequences data and sequence polymorphism. So similar to this gene bank, the GenPept is also there, and that is called the GenPept database for protein sequences. GenBank is for the nucleotide sequences, and GenPept is for the protein sequences. So you know that the gene bank is nothing but it is a nucleotide sequences for more than 300,000 organisms with supporting bibliography and biological annotations, and the data type is nucleotide sequences, and gene peptide, that is the protein sequences, and it consists the nucleotide data of all organisms. Here is some of the organisms whose sequences are submitted in the gene bank. For example, the Homo sapiens, about 1.6310774,187 into 10 to the power 10. You can see that how these huge data are submitted in the gene bank. Similarly, for the musculus and for the Drosophila melanogaster, also it is 1.11996522 into 10 to the power 9. So some of the organisms database is presented here. So let us have the credit to the first man who is behind the establishing GenBank. The GenBank, it was started in 1982 by Walter Good of Los Alamos National Laboratory, and since then it becomes an open access platform where the annotated collections of all publicly available nucleotide sequences and the protein translations data are present. And this data is produced and maintained by the NCBI, which is a part of the National Institute of Health in the United States. So, as part of the International Nucleotide Sequences Database Collaborations, INSDC. Yesterday, I also told you about that there are three databases. One is the NCBI, and another one is the DDBJ, and third one is the EBI. And these three or developers, they just unite together to form a collaborator. And that collaborator name is the INSDC. So Gene Bank and its collaborators receive sequences produced in the laboratories throughout the world from more than 100,000 distinct organisms, and then it becomes an important database for research in biological fields and has grown in the recent years at an experimental rate, exponential rate by doubling roughly every 18 months. And the current release, latest by the February 2021, the release is 242.0, and it contains about 12 trillion nucleotide bases in more than 2 billion sequences. So you can see that this graph that there was a tremendous increase of the database starting from the 1982 to you can see up to 2021 the rate of the how the data has increased. So how you can search the this gene data bank? Suppose you want to know the sequence of any genes, so you will have to go through the platform of the website. So there are two methods. Uh, this gene database can be searched by two methods. First, by using the text-based keywords similar to the PubMed search. You just open the windows and you just type the name of the genes and go search. Then after that you will get the record, or you can have the using the molecular sequences. You have the sequences, you just paste the sequences and search by sequence similarity using the blast. But here I will just confine to only the one of the method that is using text-based keywords similar to the PubMed research. So for this you can have the you can just go to the this platforms and let's just open the our website. So if you just open the website, then you can have the entire GenBank review. So in this page, you see that the upper panel it has got the nucleotide. You can there are many so many types of the sequences are there, and this window is for the searching the uh, desired genes, and here some of the descriptions are there. Here it is written that GenBank is the NIH genetic sequences database and annotated collections of all publicly available DNA sequences, nucleic acid. So, and all this they have got the hyperlink. You can go to the anyone. This is GenBank home submission type, submission tools, search GenBank, updated GenBank records. So many records are there. Okay. So this is all about the your that the how to access the gene bank. Then privacy is there because as, the developer they also have the certain privacy. So let us just go to the one of the sequences. Say how the gene database gene database they consist of the certain records, and the records have got the certain definite format, and that definite format is the computerized readable form. So let's open one sequences. For examples, you just open the your sequences like insulin. Okay, uh, insulin. The first, go to the search, and when you just go search, then you get the uh, certain sequences complete. So here you can see that if you want to know that octagon Degas insulin mRNA complete series sequences is there, and here's the gene bank. It means that in this gene bank, the record of this insulin mRNA sequences are there. So just click on the gene bank and you will get the entire records of the insulin. Okay. Now the this is called as the file format. It's the flat file format. And if you look at this flight format, then it has got the three sections. Three sections. The first is the head section. This is the head sections. This one. I'm just selecting this one. So this is the head sections, and after that, this is the feature sections. It is the feature sections, and uh, after that, the sequence section is there. Okay, it is the sequence section. So, if you just have a look on this head section, so it consists of many subheads. For example, the locus is there, locus. Then it has got the how many sequences are there. It is the 432 base pair sequences, and what the nature is the messenger RNA, and it is a linear. And the definition is there from which species it has been obtained. It is the octagon Degus. It belongs to the insulin, and the complete coding sequences are there. Then accession number. So accession number here one alphabets and followed by the 1, 2, 3, 4, 5 digits, and these are the accession numbers, and these are permanent. So, if you want to search these sequences, you can search either by the typing the keywords that is the insulin, or you can just copy these accession numbers and you paste it. You can get the sequences. Okay. Uh, so, and this is the versions. I told you that the biological database has got the uncertainty. That is, when it is submitted, it may be true or it may not be true. So some others has to validate them. When they validate, and if they found certain missings, and that missing is incorporated, and the old version is then superseded by the new versions. So it is the M56. The number never remains the same. So the accession numbers it has got the point one. So M the say before the decimal the number is same, and it is the first version. Another, if it is the second, then second two number will be there, then third number will be there. So accordingly that so after the decimal that this number represent the versions, which number of versions are there. Okay. Now, this keywords is insulin, and this insulin may have the alpha chains or the beta chains or the entire connecting peptides are there. Then organisms, and after that you can see here the classifications. Classification where this octagon, the octagon belongs to the eukaryotes. It is a metazoa. It belongs to the chordate, then craniata, then vertebrata, then etiostomali. It is the mammal eutheria. Okay, so these all are the you can say the taxonomical positions are there. Okay, and after that the references are there. References is only one. Okay, and who is the author? Money. It means that key who has submitted this database. So this database had been submitted by the Nisi M and the Steiner DF, and the title of the topic was the cloning of complementary DNAs encoding islets amyloid, polypeptide insulin, and glucagon precursors from a new world rodent, the Daegu octadon dagus. Okay, and in which journal it was published? It was the molecular biology endocrinology portion, and in 1990, and this is the page number. Okay, and this is the PubMed. So if you want to see this articles, then you can just copy this one, or you okay, you can just click. You click, you can get that paper. So okay, so this is the paper. So this is the paper of the this one. Okay. So so let us just back to the your let us go uh, back to this. So again, the after that the section is your feature section. So again, the feature section, that the locations, qualifier, quality qualifiers is there, then organism is there, molecule type. So these are abbreviation. You can also say that key what is the tissues? It is the pancreatic tissues are there. Okay, what the genes number? Gene is the 1.432. Okay, gene is insulin. Codon start from the one. Okay, and after that you can see the sequences. So this protein, it is a translational product. So you can say these are the sequences. Of course, the number is already there. Number is I think it is 432 base pair. So it is the 432 base pairs. Okay, so these are the all the detail of the zigam, and after that it ends with the origin. Origin. So this is the you can say this is a 421, 421, 22, 430, 23, 424 like that. It will be the 432. Yeah. 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32. Okay. So these are the sequences of the nucleotide, and these are the sequences of the proteins. Okay. So this features also give the key what is the protein product of the this very nucleotide sequences, and after that this ends with the two slash. So this is the slash. So it is regarding that key how the gene bank format is there. So from this format you can have the knowledge of all about the this very genes. Okay, let us back to this one slide. So you just see that okay. Now, so I was dealing with the this things. Okay, so you can see that the same things I have been uh, showing here. The gene bank sequence formatted. It has got the header sequences, feature sequences, and the sequence sections. Okay, and now you can see that this is the format what I showed you in the website, and this is the first section. This is the header part. Okay, you can see here the header part is up to here, then after the feature is there, and from the origin research sequence is there. Okay. So this line, the header section, this line by definition provide the summary information for the sequence record, including the name of the sequence, the name of the taxonomy of the source organisms if known, and whether the sequence is complete or the partial. Okay. This also provide your accession number for the sequence, which is unique number assigned to a piece of DNA when it was first submitted to GenBank and is permanently associated with that sequence. And this is the number that is cited by the cited in publications. So it has two different formats: two letters with 5 digit or one letter with the 6 digit. For nucleotide sequence that has been translated into protein sequence, a new accession number is given. If the nucleotide sequences is there, then another number. If the protein sequence is there, it is giving the another new number. Okay, and it is also having the gene index GI number is there. Okay, and then the you can say the next line is the header section is the organisms. What I told you about the octagon. Then it has also got the taxonomical classifications. Taxonomical classification is there. uh, then it is also followed by the references. And I told you about that. It is the article is available in the PubMed. It has been published by some workers when it is published, where it is published. So that the name of the journals, year of the publications, name of the authors, and the topic, that all will be given into the head section. So and the second section so that is with the feature sections, which include the annotations, informations about the gene or the gene product. So in the website, when I just logged the this insulin protein, then I got that what is the protein sequences of that very particular gene. So you can get that things in the feature section. And in the third section, the sequence is there. Okay, that is that we call them origin. Is the origins, and the section always ends with the two slashes symbols. So, what is the importance of the gene bank? The gene bank has the importance that if you want to retrieve DNA or protein sequences from gene bank, the search can be limited to different fields of annotation, such as organisms or accession number, or by authors or publication data. It means what you can have the many methods to retrieve the DNA sequences. Either you by the typing the key, or by the organisms you write to cite, or by accession number, or by authors, or by publication data. Whatever you want to write or paste the option, that is open for you to just have the gene bank file format. So it is a very easy task to just upload or download any DNA sequences belonging to any genes. So, and beside this, you can also go through the publications, and you just read out the all the publication, all the details of the disease. So it means that there are so many other formats are there. So many formats are there. Uh, um, so many formats are there, you can see here. Okay, uh, let it be. Okay, this is the so yeah, you have got the many format here. So you found that there is the this one, the article is there that has been published by for the GenBank. And after that you just go back, uh, you write the name of the any genes. For example, I am writing here trypsin, TRY. ESIN trypsin. Then go search, then you got the trypsin. Then you go to the GenBank. Okay, so here you can see in the right panels. In the right panels also it has got the another file form that FASTA that will tell you later on. Then graphics is also there. Then in this panel the run blast, peak primers, or bioproject, biosample project, whatever the things you want to just get the information regarding this very genes. So they have got the so many link. Okay, so many links are there, so you can go to these very links and you get the information. So okay, uh, and if you want to know about this, this is the trypsin, but no sequences are there. It means that the sequence are not available. Let me check here. Yeah, it has got the one. This is base pairs. Base pairs. So let me about this taxon is there. Oh, so this is the taxonomy. So by this you can just uh, in the nutshell you can have the information that GenBank is nothing but is a nucleotide sequence database.